students, welcome to the YouTube channel of YourPedia Education. Today we are going to study about a very interesting topic from environmental microbiology. The topics are growth curve and Monarchs Equation. Let's get started with it. First, we are going to learn about growth curve. Before we get deeper into this topic, I want you to be aware of these two terms. First one is growth. What do we mean by the term growth? And the second one is proliferation. These two terms would be used interchangeably throughout this lecture and they would signify more or less the same thing. So growth in technical or microbiological term means the growth of biomass of a cell population, right? And proliferation. Proliferation. It means the growth in cell number of that microbial population. All right. Both of these terms signify growth only, growth in either the biomass or the cell number. These two terms have to be in conjugation with each other. Growth should be directly proportional to proliferation for a balanced growth. That means increase in cell number should also mean that there is an increase in the total biomass of that microbial population and vice versa. And if there is any mismatch in this proportionality, then the growth that is the result is called as imbalanced or unbalanced. Or unbalanced growth. All right. So that's pretty much about it. So growth or proliferation, it takes place in two methods. Via two methods. One is linear growth. All right. We are now talking in terms of proliferation or cell number because it's mathematically easy with respect to biomass. And the other type is exponential growth. So growth curve, as we can see on the left side of our slide, it follows both these patterns, linear also as shown here and exponential also as shown here. So for linear growth, let's say, let's define a few terms here. Let n be the increase of cell concentration. Cell concentration, all right. So n is the rate of proliferation, also known as increase of cell concentration, right? And let's say the time required to double the cell number in a population can be considered as an average cell generation time, Tg. So Tg is the average cell generation time, the time taken to form new cells. All right. And then the number of cells as well as the biomass can increase linearly in case of formation of new cells on the tip of hyphae, this portion of the curve, all right, which can be defined by the equation. So this procedure follows this maths, K and naught, where N naught is the initial concentration of cells and K is the growth rate. All right, the constant that is introduced to dissolve the proportionality. And when it's exponential growth, as shown or depicted here in the log phase, this equation becomes n is equal to n naught 2 to the power n. This is the math. This is the mathematics that the microbes follow during this phase of their growth. Alright, so they follow both linear and exponential growth. Linear is a bit slow, way slower than the exponential growth, and exponential growth is the one where we get the maximum specific growth rate, which we are going to study about later in this lecture. Alright. Okay. So, from civil engineering perspective, what happens in a sludge digester or in a, let's say, aeration tank, or from a biotechnical perspective, so what happens is in a bioreactor or in a sludge digester in a chamber, where let's say our wastewater is present, we want to decompose it with the help of our microorganisms. We want to make it or convert it into simpler and less biohazardous waste so that it can be disposed of safely into the natural environment. So what we do is we introduce our microbes here or bacterias, all right, and we give them food. We want them to thrive in this environment, eat the waste material, decompose it further down to simpler complexes or simpler materials, simpler composition of this wastewater, so that we can then easily and we can then, without causing any harm to the environment, dispose it off into our, let's say, land or water bodies without causing or with causing minimal harm to the environment. Okay, so when we add microbes and the nutrients or the substrate that is required for the growth of those microbes, for the survival of those microbes, the microbes do not just right away start eating it and growing and, flourishing in that environment. They take their own sweet time to know the environment, to get used to the food that they are being fed, and then they, you know, decide whether they want to live in this reactor or whether they want to eat this food or not. And when they have decided positively, they grow in number, they grow in size, their overall weight increases, and then they, you know, flourish in that environment and they have their highest growth rates being accomplished here. Then the lifetime that is spent inside that sludge digester or bioreactor is defined as the growth curve, as shown in the left hand side of this. Okay, so what happens first? The microbes are introduced, the nutrients are added, and the microbes now enter the growth curve through lag phase. This phase is called as the lag phase. The almost flat portion of this curve is lag phase. What happens during lag phase? During the lag phase, the microbes are testing the waters. In philosophical terms, testing the environment, testing the food that is being provided to them, testing the nutrients, testing the temperature, pH, everything that affects their survival. All right. So they take time to get adapted to the environment. And all they end up doing during this phase is just growth in cell size. So the size of cells increases in lag phase. Because the size of cell is increasing and not the number of cells do not increase. Only the size increase. So there's a very slight increase in the total biomass also. slight increase, right? So during lag phase, the cells increase in size. The microbes are getting adapted to the environment, right? Then, after they have decided that they like the environment, they like the food that is being provided to them, they enter the so-called log phase. Log phase is the exponential phase, this one. Because the growth that takes place here is in the order of exponentially. And here the growth is linear. Okay, here the growth is exponential. What happens here is the growth rate here is greater than the death rate. Way greater than the death rate. All right. And the microbes are enjoying the environment, enjoying the food that is being provided to them. And the biomass is growing like anything in this phase. The only limiting condition here is the availability of nutrients. Because the microbial population is growing so fast here, they get competitive with each other for that food. And because every biological process creates materials that gets expended in the end also, expended in the end in the form of waste material. Here also that waste material is produced, and those are toxic in nature, so toxins are also produced here. So these two are the limiting factors in this phase. So as long as the nutrients are available for the growth of microbes in the log phase, the microbes will stay in the log phase, stay in this phase. And as soon as the level of toxicity that is desirable, or that is the the level that the microbes can handle, as long as it's within the limits, the microbes will stay in this phase only. And as soon as the threshold is crossed, the level of nutrients drops and the level of toxicity increases, they enter the so-called stationary phase. In stationary phase, the growth rate in stationary phase, the growth rate is equal to death rate. The growth rate of the microbes is equal to the death rate. The number of cells that are being produced in this phase are equaling the number of cells that are being destroyed also, that are dying also, right? So there's no growth, or we'll call it, there's an equilibrium acquired or reached here. In this phase, toxins are increasing, and there's a formation of certain metabolites, which we call as secondary metabolites. In log phase, the one which we studied previously, primary metabolites were formed. These metabolites are important with respect to biotechnological view. So the secondary and primary metabolites are formed in stationary and log phase respectively. Under primary metabolites, we get amino acids, other organic acids, vitamins, alcohol, and acetone. So from, let's say, pharmaceutical point of view or industrial point of view, if we want to generate or produce any of these amino acids or vitamins, alcohols or acetones, let's say in case of breweries, we keep our bioreactors running in the log phase to get these products. And under secondary metabolites, we get antibiotics. We also get steroids. So these are important from the biotechnological point of view, not the civil engineering. So these are the metabolites which are formed in stationary phase and in the log phase, primary and secondary. Right. After the stationary phase, we enter so-called death phase. Death. In the death phase, the growth rate drops and it gets lesser than the death rate of the microbial cells. This death rate is also exponential. In log phase, the cells were growing exponentially. In death phase, the cells are dying exponentially. All right. And this marks as the end of growth curve for the microbes in a container or in a bioreactor or in a sludge digester. This is the time during which this whole process from the start of or from the birth of microbes to the death of microbes takes place. And it's always plotted on the growth curve is plotted on the semi-log scale. On the y-axis, we have number of bacterial cells or the biomass concentration in log form, which could be natural log or 10. All right. And on the x-axis, we have time duration, time taken to complete this whole life cycle of the microbe. The growth curve is nothing but the life cycle analysis microbe. So microbes first enter the lag phase, then they grow and enter the log phase, then they enter the stationary phase, and then the death phase. So all these properties that we just discussed are enlisted here. What happens in lag phase? Cell numbers remains constant, yes, because they only increase in size. And biomass is almost constant. There's very slight increase in biomass because of the increase in the cell size. In log phase, the cell numbers increase exponentially of the order 2 to the power n, right? And biomass also increases proportional to that. And the limiting factors here are nutrients and toxicity. The level of toxins which are present or produced during this phase. The production of primary metabolites takes place in log phase, and the ones which are produced here are amino acids, organic acids, vitamins, ethanol, and acetones. In the stationary phase, the cell number again comes out to be constant because the growth rate equals now the death rate. The toxicity increases to very high alarming levels, and there's a production of secondary metabolites, which are antibiotics and steroids. And the last phase of the growth curve is the death phase, where the cell number decreases exponentially and the death rate is much, much greater than the growth rate. All right. Now we have been talking constantly about growth rate, specific growth rate I mentioned maybe for 3 times. So let's know what it actually is. Okay. So let's say in the start, we had fed around 40 grams of microbes, which doubled in 10 minutes to 80 grams biomass, right? And let's say in another bioreactor, we took the same microbes, same bacteria, but we started with 80 grams, which again doubled in 10 minutes to 60 grams. So growth rate and specific growth rate are two different terms which talk about the rate of growth. So what is growth rate? Growth rate is increase in the biomass per unit time. So as per this definition, for case 1, this case 2. For case 1, the growth rate comes out to be, what is the increase in biomass? It's 80 minus 40, that is 40 divided by 10, that is 4 grams per minute. All right. And for case 2, we get 160 minus 80, that is 80 divided by this 10. So this 8 grams per minute. So there are two different growth rates for the same type of microbes, given that the initial conditions are different. Okay, the growth rate is depending upon the initial amount of biomass that is fed to the bioreactor. Now let's see what specific growth rate gives us. Specific growth rate is nothing but growth rate per unit initial biomass. So for case 1, it would be, what was the growth rate? 4 grams. And how much was the initial biomass? 40. It comes out to be 0.1 per minute. Right? And for case 2, what is it? It is 8 divided by, what is the initial biomass? 80. It again comes out to be 0.1 per minute. So here is the difference. We see that specific growth rate remains same for a particular type of bacteria or a microbe, while growth rate is different. It depends upon the initial amount of biomass. So that is the main difference between growth rate and specific growth rate. Specific growth rate is independent of the initial concentration or initial amount of biomass that is present in a bioreactor, while growth rate depends upon it. And hence, their units are also coming out to be so. For growth rate, it's grams per minute. And for specific growth rate, it's per unit time or per. All right. So we can get to know the type of bacteria by just looking at the specific growth rate, but we'll not get any idea about the type or the characteristics or the properties of particular microbial population by looking merely at the growth rate. So specific growth rate gives us a better idea about the type of microbe, and it is independent of the initial biomass, right? Specific growth rate is found out from the exponential part of the growth curve. Growth curve looked like something like this. The slope of this portion, slope of this gives us specific growth rate, which is depicted like this. Mu, or specific growth rate, it's represented as mu at any time t is equal to ln x dot v, cell concentration in log form, natural log. This is ln x dot v point. This point is ln x naught dot, right? And the perpendicular, the difference of this and this will give us perpendicular, and this time duration will give us the base. So as we know, tan theta, let's call this tan theta, is equal to mu, our specific growth rate, which is perpendicular upon base, which is this term upon this term. That is how we get our specific growth rate from the slope of exponential portion of growth curve. In very general science terms, specific growth rate is defined as the grams of biomass built per gram of biomass present per hour of the process time. Okay? Biomass per gram of biomass is per unit time is what we call as specific growth rate, right? So, what are the properties in view of this specific growth rate of the growth curve? During our log phase, the specific growth rate is 0. All right? Because there's no growth taking place. There's only increase in size, cell size. So, specific growth rate is 0. As we can see, the slope is 0. It's flat. Hence, it's only natural, or let's say it's only mathematical, that the mu is 0 in the lag phase. The next one is the log phase. During log phase, I have written it ulta. During log phase, the specific growth rate reaches its maximum. Okay? During stationary phase, it's almost equivalent to 0, because growth rate becomes equal to death rate. And during the death phase, which is the negative growth, or the where the death rate is way greater than the growth rate, the specific growth rate falls below 0, less than, right? So during lag phase, it's 0, it's maximum, it's again 0, falls below 0, right? Now let's study about Monod's equation. Monod's equation is nothing but it gives us an idea. It's one of the equations, one of the mathematical connections between how much substrate should we provide to our bacteria for them to achieve their maximum specific growth. We just want to know that how much amount of substrate or nutrient should we provide to the microbial population that we are dealing with in a bioreactor or a sludge digester for them to achieve their maximum possible specific growth rate. And one of the equation which gives us this relation or an idea about this is Monod's equation. Monod's equation is derived from its primary form, which is also known as Michaelis-Menten equation. About that right now. Just know that it's derived from Michaelis-Menten equation, and it gives us a relationship between specific growth rate and the substrate concentration. So, what is this equation? The equation is specific growth rate at any time t is equal to specific maximum growth rate, maximum specific growth rate into the substrate concentration at any time t divided by the substrate concentration plus Ks. Ks is a term which means it's the substrate concentration. Here, look at it, this graph. Substrate concentration at which half of the maximum specific growth rate is achieved. Let's say the maximum specific growth rate is 10 per minute. All right. So when 5 per minute is achieved for a particular microbial population, the substrate concentration at that point of time is Ks. It has the same units as that of substrate concentration. As the law of physics says, For two quantities to be added to each other, they must have same units. So S is being added to Ks. So Ks has to have the units of S. All right. And from this graph, we can observe that there's only one particular point, one particular amount of substrate for a particular microbial population that we can provide to achieve the maximum specific growth rate. After that, no matter how much amount of substrate you keep on providing to your bacterial or microbial population, there would be no increase in this specific growth rate. It will cease to be increasing, and because this curve goes flat after one point of substrate concentration. So we just need to find out this concentration to achieve this maxima. All right. After that, there is no point of adding extra substrate to our microbes because that is not how they are related. Giving more food to our microbes will not ensure more specific rate. It has its limits, which is decided by this mathematical. So let's see what all these parameters and variables signify. Umax is the maximum specific growth rate. Mu is the specific growth rate at any particular time. Ks is the substrate concentration at which the half of the maximum specific growth rate is achieved. And S is the present substrate concentration at which we know the specific growth rate. All right. Certain properties of Monod's equation are enlisted here. First of them is that the unit of Ks is same as that of substrate concentration, as I mentioned previous slide. The next one is Ks is inversely proportional to the affinity of microbes to the substrate that is provided. What this means is substrate or what this means is that Ks value for different types of substrate is different, and a higher Ks value shows that higher amount of substrate is required to achieve half of the maximum specific growth rate, which means that the microorganism has a lower affinity for the substrate. What it says in simpler terms is that the simpler the food that we provide, or the most favorable food that we provide to our microbial concentration, they will take lesser amount of time to eat it, and lesser amount of time will ensure that Ks is reached faster, right? So good food, favorite food, faster Ks. So that means if amount of Ks is less, affinity is more. That means faster reaching of Ks means lower amount of substrate that is left undigested or left undigested by the microbes. That means the bacteria are liking the food. That means they have higher affinity. Higher affinity means higher liking towards that food. So Ks is inversely proportional to the affinity of microbes to the substrate. That is what it means. Again explaining, whereas the lower Ks value shows that even a low amount of substrate concentration is able to achieve half of the maximum specific growth rate. Hence, that microorganism has a higher affinity for this particular type of substrate. These two statements say same thing, that Ks is inversely proportional to the affinity of microbes. Alright? It has been found that if the favorable substrate is not provided, or the substrate provided is complex in nature, the microorganisms find it hard to break down that substrate, and hence the maximum specific growth rate may decrease. Okay. So the simpler the food, the tastier the food for the microbes, the more will be the maximum specific. And Monod's equation can be used as it is for the calculations regarding batch culture. So there are different culturing techniques: batch culture, fed batch culture, and the continuous cell batch culture, in which we utilize the microbial populations for our benefits, like to produce primary or secondary metabolites. And Monod's equation is one of the equation which can be used directly for calculations with respect to the batch culture. So that is all about the growth curve and the Monod's equation. I hope you guys have taken a few points to understand and remember this from my lecture. Thank you.